In this module of today's lecture, I want to go into a little bit more of the advanced biology that's involved in plant signaling or in plant responses to light. I'm going to assume a little bit knowledge of basic biology, and I'll try to explain some of the more advanced concepts. So earlier in today's lecture, we talked about and we learned about how a plant responds to blue light, how there's phototropism, the plant absorbs the blue light and bends towards that signal. But we didn't really talk about what's involved in the signaling, how the plant is responding to the blue light. Several decades ago, it was found out or was discovered that one of the first things that happens when a plant is illuminated with blue light is a change in a specific protein. One protein in the membrane of the plant cell becomes phosphorylated. What that means is that phosphate is added to this protein. The way we can check this, the way that scientists check this, is that we actually add radioactive phosphate to the plants, isolate the proteins, and then see which proteins have become radioactive, which have absorbed the radioactive phosphate. What we can see here in this picture, this is actually showing a specific protein that when it's in the dark, it has a certain level of phosphorylation, and we see that by the width or the size of this band in the gel. And here, the same protein, after it's illuminated with blue light, it gets much stronger signal. That means there's a lot more phosphate on this protein. And this is induced only by the blue light. Okay? So here we have a protein that becomes phosphorylated in response to blue light. Now, in all of biology, phosphorylation is one of the ways that proteins signal to each other. And this is true for all of animal biology, all of bacterial biology, and all of plant biology. We also learned a little bit about the mutants that were isolated that were blind to blue light. And it actually turns out that there was more than one mutant that were blind. Now, these mutants were called NPH at the time for non-phototropic. There were actually 4 mutants isolated. Here we could see the normal, the wild type, which is bending to the light, which is coming from the side here. And mutations in 4 different genes, NPH1, NPH2, NPH3, and NPH4, are all blind to the light. These are mutants in different genes, but they all look the same. They're colorblind. So now the question could be, what happens to these mutants in terms of phosphorylation of this same protein? In other words, if I give the blue light, is this same protein still phosphorylated? And in this experiment, we can see that the results are different depending on the mutant. For example, in NPH1, we can clearly see that there is no change in phosphorylation in response to the light. Again, I'll go through this slowly. In the wild type, we see that when it's illuminated with the blue light, there's a huge change in the size, in the amount of this band, a lot more phosphorylation. In NPH1, a mutant that's blind to blue light, there is no phosphorylation. But on the other hand, in mutants in the other mutants, in NPH2 or NPH3 or in NPH4, this protein is still phosphorylated. Meaning we're seeing a difference. We have 2 classes of proteins, 2 classes of mutants. One mutant, NPH1, affects the original phosphorylation. Whereas mutants in the other subunits, the phosphorylation still occurs, but the plant is still blind to the light. So if I fast forward ahead now 10 or 20 years, where we've now cloned the genes, my colleagues have cloned the genes, we know what they're doing, here's what's happening. So if we now take all the data that's accumulated for these phototropic mutants, here's what we now know. This is our paradigm right now of blue light signaling for phototropism. We now know that NPH1 encodes a protein which we now call PHOT1 or phototropin-1. This is the blue light receptor necessary for phototropism. When PHOT1 absorbs blue light, it becomes phosphorylated. A phosphate group is attached to it. This is the first step in the blue light signaling. Once PHOT1 is phosphorylated, it can then interact with NPH3, physically touch each other within the cell. And then NPH3 somehow takes the information from PHOT1 and transduces this signal further into the plant. The signal from NPH3 then affects a plant hormone that's called auxin. We're going to learn more about auxin later in this class, but auxin is one of the main hormones that is important for phototropism. The auxin signal is then modulated by NPH4, which itself is a transcription factor. What that means, this is a protein which directly affects which genes are turned on and which genes are turned off. And this then finally leads to phototropism. So we could see that starting from a simple genetic screen, which isolated various mutants that are all blind to blue light, we isolated a mutant in a photoreceptor. If this photoreceptor doesn't act or it doesn't exist, the plant is blind to blue light. We isolated one of the proteins involved in the signal transduction, in the transfer of the signal after light perception. If this signaling component is missing, then you don't get phototropism. And we also isolated a protein directly involved in the regulation of which genes are turned on or turned off, which would be very far downstream in the pathway. Also, if this doesn't exist, the plant would be blind to blue light. So we can affect light signaling at different levels, at the level of perception, at the level of transduction, or at the level of the response. Just like in humans, we could be blind because of a problem in the photoreceptor, a problem in the transduction in the nerves, or a problem of the response in the brain. All three defects lead to blindness, but they're caused by different problems. One last thing here. So we talked about blue light and phototropism. We briefly talked about blue light and photomorphogenesis. But blue light affects many, many processes in the plant. I said phototropism, but it also affects when plants open the small holes on the leaves, which are called stomata. It affects how chloroplasts, the part of the plant that does photosynthesis, actually move within the cell. And it affects how leaves follow the sun during the day. In other words, blue light responses maximize the ability of a plant to carry out its photosynthesis. Now, this thing about solar tracking is very important to me from a personal level, because the first class I ever learned in light signaling was taught by Professor Dov Kohler at Hebrew University, who specialized in understanding how plants change the position of their leaves in response to the direction of the sun. Recently, a book was published called The Restless Plant that Dov wrote, and it was because of this class that I took with Dov Kohler that I started studying plant responses to light. And the connection to this specific class is that Dov Kohler is the father of Professor Daphne Kohler, who was one of the founders of Coursera. So if you're interested in learning more about the mechanisms by which plants respond to light and move, I highly recommend that you read this book. So that ends today's lecture. Next week, we're going to be talking about what plants smell.